Assalamu alaikum. Welcome to my channel Pharmacology Made Easy. Many days later, we came again into the videos of pharmacology lectures, and today's lecture is all about insulin. And in these lectures, we'll discuss about the structure of insulin, how insulin is synthesized, how insulin is secreted, the regulation factors responsible for insulin secretion and insulin inhibition, then mechanism of action of insulin and how insulin controls our blood sugar level. So before discussing the insulin, we first learn about the pancreas. Pancreas is an endocrine hormone and also exocrine hormone, so we can call it's a double gland because it has two important part. One is exocrine part and another one is endocrine part. The most function of pancreas is in its exocrine portion. The 99% functioning pancreas acts like exocrine gland because it secretes many digestive enzymes and bicarbonate that are secreted mainly into the second part of the duodenum, not into the blood, and that helps to digestion of different proteins and lipids in our bodies, and also uh, release of bicarbonate that also helps in different situation of our metabolism. The endocrine portion of pancreas acts only 1%. The only 1% functioning of endocrine pancreas is mainly responsible for its islets of Langerhans cell. These islets of Langerhans cell are the cluster of the cells which is scattered throughout the pancreas. So in this endocrine part, or these islets of Langerhans are composed of four important types of cell. One is alpha cell or A cell, another one is B cell or beta cell, another one is delta cell or D cell, and another one is PP cell. It also releases another cell that is called the F cell, or we called it epsilon cell. The alpha cell or A cell secretes glucagon, and which helps in the time of starvation by glycogenolysis and gluconeogenesis. It produces the blood sugar level, it increase the blood sugar level and increase the blood glucose during the time of starvation, and that is the function of alpha cell that secretes the glucagon. The important cell that will be discussed today, that is the insulin, which is secreted by pancreatic beta cell that is released from the islets of the Langerhans, and this pancreatic beta cell composed of 60% of the islet of Langerhans cell mass. The total islets of Langerhans is composed of four types of cell, in which the main cell is the beta cell because it is composed 60% of the islets of Langerhans, and it secretes the important hormone that control our blood sugar level, that is insulin. The beta cell or B cell not only secrete the insulin, it also secrete proinsulin, the connecting peptide, and also an important hormone that is amylin. The amylin, importance of amylin is in type 2 diabetes mellitus. Nowadays the pancreas is deposited by this amylin, and it produces amyloidosis in the pancreas, and which causes the patient into amyloidosis, a disease that that uh, that involved in type 2 diabetes mellitus, and the causes of one of the cause of type 2 diabetes mellitus is amyloidosis, that is deposition of amylin into the pancreas. Okay, another cell, that is the D cell or delta cell, that is secret the somatostatin. The somatostatin, it is a universal inhibitor because it inhibits the many hormones in our body. It not only inhibits the insulin, it inhibits the glucagon, it inhibits the prolactin, it inhibits the many types of antiropituitary hormone, it inhibits the secretin, cholecystokinin, and also gastrin. So this is the somatostatin. And another hormone that is released by pancreatic polypeptide, that is the PP cell, that is called the pancreatic polypeptide. And today we will not discuss about the pancreatic polypeptide, so we are not concerned about it. Another cell of islets of Langerhans that secretes the that is called the F cell, that secretes the epsilon. Today is concerned about the insulin. So now we'll discuss about the insulin. Insulin is an anabolic hormone that helps to control the blood sugar level in type 1 diabetes mellitus. In type 2 diabetes mellitus, where oral anti-diabetic drug cannot control the blood sugar level. In case of diabetes mellitus with complications, diabetes mellitus with different type of surgery, and also an important electrolyte imbalance, that we use the insulin, that is called the hyperkalemia. Insulin is the only hormone that controls the blood sugar level, that decrease the blood sugar level, and many other hormones that increase the blood sugar level. So the structure of the insulin is synthesized from the rough endoplasmic reticulum of the pancreatic beta cell, and this rough endoplasmic reticulum from which the ribosome, the synthesis of insulin, it is processed and packaged into the Golgi complex and stored into the secretory vesicles. And due to stimulation of some stimulus, that insulin is released from the pancreatic beta cell. Now the structure of the insulin. Insulin comes from different precursors. The first, the insulin precursor, the name of precursor of insulin is pre-proinsulin. It is composed of 110 amino acid, and it is composed of initial segment, the B chain, the A chain, and B chain and A chain are connected by C peptide. After that, the initial segment is breakdown, and it is composed of B chain, A chain, that is connected with C peptide. And when the disulfide link is in between them and make a bridge between the B chain and A chain, that is called the proinsulin. The proinsulin is another precursor of insulin. Then this proinsulin is break down into the insulin and the connecting peptide, and this insulin is composed of 51 amino acid. The A chain contains 21 amino acid, the B chain contains 30 amino acid, and they are linked by disulfide linkage. And another connecting peptide that is breakdown from the proinsulin, that is called the connecting peptide. And this connecting peptide and insulin are released from the pancreatic beta cell in equimolar amount. This is the structure of the insulin. Now the synthesis of insulin. Insulin is synthesized from the rough endoplasmic reticulum of the pancreatic beta cell, and the rough endoplasmic reticulum, the insulin is present. The insulin gene, if we start from there, the insulin gene is present in the short arm of the chromosome 6 of pancreatic beta cell, and this short arm of the chromosome 6 in the nucleus of the pancreatic beta cell, the DNA sequence is converted into the pre-mRNA. Then it is converted to mRNA, and mRNA helps to synthesis the protein, and this protein is synthesized into the ribosome, and this ribosome, when binds to the endoplasmic reticulum, that forms the rough endoplasmic reticulum from which the insulin is synthesized. Then insulin is processed and packaged into the Golgi complex, and then it is stored into the secretory vesicles. In the pancreatic beta cell, all people in the world that contains 8 milligram of insulin, that is equal to 200 international unit of insulin, and only 25 to 30%, so only 30 to 40 international unit of insulin that is secreted daily. Insulin is released from different types of stimulus, and the stimulants that increase the secretion of insulin are: one is glucose, and another one is high amount of glucose if we intake, the sucrose, the fructose, the increased amino acid intake. If we intake high protein food, that will be help to stimulate the pancreatic beta cell to release the insulin. If we intake high amount of fatty acid by taking lipid containing food, this stimulate the pancreatic beta cell to release the insulin. Also, the incretin mimetics, that's a hormone that produce the glucose induced insulin secretion from the pancreatic beta cell. It also helps to secrete the insulin. And these are the important stimulus that causes the release of insulin. Another important thing, that is the beta adrenergic activity of some drugs that also secrete the insulin. So these are the important stimulus to secretion of insulin, or that stimulate the pancreatic beta cell to release the insulin. The inhibitory factors that inhibit the insulin secretion, that's are low containing short chain fatty acid. Then the insulin itself inhibit the insulin secretion. The hypoglycemia inhibit the insulin release. Another condition, that is the some drugs can also inhibit the insulin release, that is diazoxide, that is colchicine, that is uh,